நீங்க செக் பண்ணுங்க. 5 5 5. ஓகே. Just a minute.
A very good morning to one and all. Today's poster presentation is on our project proposal that says vectors and asymptomatic carriers of viral pathogens of economically important crabs and shrimps. Before starting the main presentation, I would like to give an overview of the economics or the commercialization that has undergone, that the shrimp industry or the shellfish industry has undergone in the past 2 years. As we can see here, that in terms of quantity, the metric ton that we observed in 2009 to 2020 year, it was 6733 metric tons, while in 2020 to 2021 it was, the amount considerably went down to 5489 metric tons. So we observed a growth percentage of negative 18.48%, and also in terms of income in the year 2019 and 2020, the production in terms of dollar was $78.62 million, while in 2020 and 2021 it was only $54.26 million. So here also we observed a steady fall of 30.99%. So with this I'm going to start the main presentation, that is what is causing these losses in the aquaculture sector. So first I would like to say what is a vector. A vector is an organism that carries the viral diseases or the pathogens or whatever symptoms that are there from the source of the infection to the animals. So that is what a vector is. And an asymptomatic carrier is, it is affected by the disease but it does not show any kind of symptom. For example, a few years back we had asymptomatic corona patients who were infected with coronavirus but they did not show any symptoms. Similarly, what we can see here is, this is a gooseberry barnacle. What we can see here is these small nodule-like structure that are attached to the barnacles. These are the main, these are the barnacles that are attached to the crab gills, sir. So they are the asymptomatic carriers. They only carry the virus. So the two main viruses that we are dealing here is white spot syndrome virus, which belongs to the viscovirus, also known as the viscovirus family, belonging to the family of the Pneumaviridae. It is a double-stranded DNA virus with a diameter of 80 to 120 nanometer and a length of 250 to 380 nanometers. And another virus that we are dealing here is mud crab reovirus, belonging to the family of Reoviridae. It is a double-stranded RNA virus, sir, and the diameter is 70 nanometers, and the site of infection here is gills, hepatopancreas, and the connective tissue. Other name for this mud crab reovirus is also known as sleeping diseases. So the key objectives of our project work was first to study the incidence of the WSSV or the white spot syndrome virus and MCRV, mud crab reovirus, on the carriers and the vectors organisms in the grow-out ponds all along the coast of the all along the east coast of India. And the second objective was to study the infectivity potential of the virus through different routes that we did in the laboratory. And the third objective was to experiment and analyze the virus and the host relationship so that we can find some preventive measures to this disease. So the methodology that we adopted was it was a 5-step methodology. Initially we did sample collection. We did sample collection for both the vectors and the asymptomatic carriers as well as the infected animals. Mainly we did sample collection all along the east coast of India. After bringing the sample to the lab, we carried out the histopathological studies where we found out that inclusion bodies are present, and we could study that the animals were infected with diseases, and then we did the ultrastructure study to understand the structure and the shape of the virus, whatever that is affecting the animals. Then we did infectivity studies in the laboratory, that how it is getting infected, how the animal is getting infected, through what route it is getting infected. Finally, all the infected animals were again undergone through molecular analysis, where we did the confirmation studies for the presence of the virus in both shrimps and crabs. So for preliminary work, what we did is we collected samples from farms and landing centers. These are mud crabs, Scilla olivacea and Scilla sedata. As the preliminary studies, as I mentioned earlier, we did histopathological studies. We collected, as I mentioned earlier, the site of infection is gills, hepatopancreas and connective tissues. We collected all the tissues, we stained them, and we did solidified them in wax blocks, and we undergone histopathological studies where under the microscope we could see these inclusion bodies, as we can see here, this hyperatrophic inclusion bodies, where we can see the enlarged bundle-like structure within the connective tissues of the crabs. And then we went for the ultrastructure studies of these inclusion bodies. Once these inclusion bodies were done and ultrastructure studies were carried out on these inclusion bodies in a transmission electron microscope, we could see that they were rod-shaped viruses with the length of 200 to 300 nanometers. And further, the molecular analysis, that 941 base pair, we did it with specific we did the molecular studies in gel electrophoresis system, which confirmed the presence of the virus. Similarly, we carried out for WSSV also, we did shrimps also, sir, not only crabs. So two commercially important shrimps that are prevalent in the east coast of India is Peneus venami and Peneus monodon. Monodon is also known as tiger shrimp. Venami is Pacific white-legged shrimp, sir. We did the same for them. We did histopathological studies, ultrastructure studies and molecular analysis, where the same 941 base pair was observed in the molecular analysis, which confirmed the presence of WSSV virus. For mud crab virus also, we collected mud crabs and we did histopathological studies. Here we can see the margination of this chromatin, these basophilic inclusion bodies, where the inclusion bodies are too much granular, which is why we call that. In the ultrastructure studies of the hepatopancreas, we saw that 70 nanometer icosahedral, it is like a honeycomb compact structure, which is why it's known as icosahedral shape or the non-enveloped shape. We saw that no spikes were there. As we see in common viruses, the viruses are spiky in nature, but no spikes were observed in the ultrastructure studies of mud crab virus. And for the molecular analysis, 433 base pair, it confirmed the presence of the molecular studies confirmed the presence of the mud crab virus. Now the main missing link to the research is what is more affecting, what is creating the disease more, the vectors or the asymptomatic carriers. The vectors, as I said earlier, they are the carriers of the diseases, but asymptomatic carriers are themselves affected by diseases. So through this study, it is unclear that what are the main causative agent of both the diseases, either the vectors or the asymptomatic carriers, since both are prevalent in the culture pond as well as the wild environment. So the expected outcomes of the study is, through this study we can do the diversity check of the WSSV and MCRV on all along the east coast of India, and we can do the pathogenicity also, how they are affecting, how they are doing loss to the culture facility and everything. Through this we can tell the farmers, we can enlighten them about what precautions to be taken, because unlike human beings, shellfishes or crustaceans, they don't have a memory system, which is why viral diseases cannot be cured in case of shellfishes. Whereas in case of humans, we see viral diseases can be cured with certain medicines, but for shellfishes, if they are affected by any kind of viral diseases, they die. So for MCRV, what we are seeing is mud crab virus, from the day of stocking, it takes 30 to 35 days maximum. Within that, mass mortality is observed. And for WSSV, white spot syndrome virus, 7 to 10 days complete mass mortality is observed, sir. So that is why it is very necessary to enlighten, to impart knowledge within the farmers what precautions to take during the stocking, which is why this study will help us to do that also, sir. So with this, I would like to end my presentation here. Thank you so much.
Selected so far?
So we selected mainly mud crabs because
reason behind this?
So mud crabs are robust in structure. They are larger compared to the marine crabs. Marine crabs, only in abroad we see snow crabs or king crabs are there. They are larger in structure. But in Indian subcontinent, all the marine crabs that are available are compared.
Is it available in our coastal area?
Yes, sir. Yes, sir. Sir, the mud crab virus that I talked about, it was first recorded in China. Then the first report was reported by our center in a crab farm in Cuddalore, sir. So after that, we moved our studies to further north, that is West Bengal side, West Bengal, Orissa, Andhra. Then now we are planning to shift to the west coast also. So we have not yet, but we are planning to shift to the west coast also.
Reason why you are planning to shift there?
Because since we found it in the east coast, that is why we want to check whether in the west coast it is available or not. because as I shown in the very first slide, sir, that here, sir, compared to 2019 and 2020, sir, we observed a steep fall in the production as well as in terms of metric tons and in terms of dollars also we observed a steep fall. This is the reasons, and as I mentioned earlier that with mud crab reovirus within 30 to 35 days complete mass mortality will be there, and WSSB within 7 to 10 days complete mass mortality will be there. So once the disease is set in the farm, within 7 to 10 days the entire farm will be emptied, all the animals will die, complete mass mortality and heavy losses will be there. As we can see here, sir, in 2019 to 20 the metric ton production, and in 2020 to 21 the metric ton production. So this is what, sir, it was first reported in Tamil Nadu after China. Yes, sir.
What is your final objective, what you are going to be developed now?
Sir, the preventive measures, the prophylaxis.
Preventive measures?
Yes, sir. Preventive measures for both the viral diseases, because as I mentioned earlier that shellfishes don't have a memory system.
Okay.
Unlike humans or other higher level of animals, they don't have a memory system. So once they are set with any sort of viral diseases, they will die eventually. But what the farmers can do is they can do prophylaxis or preventive measures. In order to prevent the disease from happening, they can take certain steps or certain precautions like fencing, biosecurity measures like fencing around the farm, or maybe some bird will fly over the farm, they can drop something that the beatings also might contain the virus. So bird fencing, biosecurity measures the farmer has to take, and there are certain medicines, certain probiotics and prebiotics that can be used to make the water sterile and free from any kind of microorganisms.
From the Department of Genetics and Plant Breeding, doing research under Dr. R. Narayan. Sufficient more microbials, they contributed my work. My research topic is on enhancement of saline tolerance in rice unit through innovative breeding approaches. The basic need of any human being is food, shelter, and clothes. Rice provides more than 50% of food for the world population, so rice contribute major in food industries. So, majority of the human population depends on rice cultivation because it acts as the livelihood for more than 50% of the human population in the world. You can clearly see the global land area, which is about 57 billion square kilometer. Currently, rice is cultivated in an area of 130. Currently, rice is cultivated in an area of 165 billion hectares with a production of 515 million metric tons. In India, it about 45 million metric tons. For 45 million hectares, the production of 130 million metric tons. In the year 2050, the demand for food will grow up to 140 million metric tons. And that too, we have to produce available land area. But the current area of available cultivable land is reduced. In 2050, it is expected to be reduced 10% from current land area. Constraints in rice cultivation. Because of human activities against the nature, there is major problem in cultivation of crops. Main thing is climate change. Because of human activities against the nature, climate change happens. Here, which cause stress in crop plants, especially stress can be classified into two types: biotic and abiotic. In abiotic drought, major stress is drought. Then next one is saline and flood. In saline, stress can be classified into halophytes and glycophytes. In halophytes, crop can be grow under maximum saline condition, which allow to grow on saline condition. But in glycophytes, crops cannot grow on this condition because they are susceptible to saline condition. So, majority of the cultivated crops coming under glycophytes, these crops cannot withstand higher salinity. So the problem is due to climate change, the increase in salinity causes major cultivation problems. rice also coming under the category of glycophytes. Here you can clearly see the response of rice to salinity. In early stage, germination and the emergence of radicle and plumule. At this stage, the seedlings were tolerant to salinity, but in seedling stage and the activity level stage, they are somewhat tolerant. But in flowering stage, it is mostly susceptible because rice is susceptible in seedling and flowering stage. Here you can clearly see the normal growth of seedlings and rice crop grow in other stages, but in seedling and flowering stage, it is maximum saline susceptible. The objective of the study is to identify saline tolerant genotypes and high yielding tolerant genotypes. From the selected tolerant and susceptible genotypes, we can find a new variety which is going to grow in extreme saline condition with high yielding capacity. We have used 48 rice genotypes. The methods we have both for laboratory methods and the greenhouse. The methods we have used is laboratory and greenhouse screening and field evaluation. In laboratory screening, saline hydroponics and molecular analysis were done. In field evaluation, quantitative traits were measured. In the field evaluation, we have observed 10 traits, and in the saline hydroponics screening, we have measured 7 physiological traits. In molecular screening, 45 SR markers were used for this study, out of which 34 gives polymorphism. From them, 10 were saline tolerant genotypes, which will confirm the presence of saline tolerance in tolerant genotypes, and 24 were yield linked markers. These are the components of organic solution which we have used in hydroponics screening. Here the seedlings shows some response to salinity. First, the scoring index 1 shows healthy seedlings under the normal and saline hydroponics. Here the EC is more than 7. We have used the artificial induction of salinity through sodium chloride. Here the scoring index 1 shows the tolerance of seedlings at EC7. In scoring index 3, it shows some susceptible expression, like leaf tip shows whitish color, and in scoring index 5, the seedlings is almost stunted growth with very much reduced in leaf area. In scoring 7, the seedling is almost in the condition of dying stage, but in scoring index 9, it is almost dying. Based on this, we have selected the tolerant genotypes. Here you can clearly see the electrical conductivity EC4 and EC7. EC4 denotes the presence of salt which is measured at 4. In EC7, the salinity is more. Salinity is measured in terms of electrical conductivity. So here in EC4, we can clearly see the well-developed root growth and leaf growth. But in EC7, the root growth is very much reduced. Because of salinity, it shows it adversely affects the seedling growth at the seedling stage. We have conducted the evaluation in pot and field level. From field, we have selected 6 susceptible high yielding rice genotypes, and in pot and in hydroponics, we have selected one. From them, we have evaluated in the pot, that is the EC is 5.5. Compared to field condition 2.8, the EC in the pot is 5.5. That we can observe 55% of yield loss in susceptible genotypes compared to tolerant one, which is lost 15 to 20% in high saline condition. Here in EC13, only tolerant seedlings were produced the leaves. The susceptible were completely died or are not able to withstand the saline conditions. The field, the observed characters were subjected to statistical analysis using identical T-square statistics. Here we have found that they were distributed in 8 clusters. The cluster distance, which shows the genetic variability present in the genotypes. Here 22 denotes these clusters have minimum genetic variability. In cluster 8, which shows maximum cluster variability. From them, I have selected the genotypes. Here we can clearly see the mean values of the genotypes present in the clusters, 8 clusters. We can select the genotypes from cluster 1 and 2, which shows maximum yield and the number of grains per panicle. Cluster 1 and 4 shows maximum grain values, which shows these genotypes can produce more grains in hybridization followed by selection. Here we can clearly see the genotypes. From cluster 1 we have selected 4 genotypes, from cluster 2 we have selected 3 genotypes, and in tolerant genotypes present in cluster A and cluster B. These genotypes were replaced for hybridization program and further selection. Conclusion. We have selected 6 genotypes from high-yielding susceptible to field evaluation and saline hydroponics, and based on saline hydroponics we have selected 4 tolerant genotypes: epicursanate, Hocali and CSR10, CSR36. These genotypes shows maximum tolerance towards salinity. So the tolerant genes were transferred to susceptible genotypes through hybridization program. So we can easily develop the high-yielding variety from hybridization. Future research. We have so far selected the parents for hybridization program. Further, we are going to conduct hybridization followed by selection program in the further seasons. Advantages. We have utilized mortar for this study, so we can develop the crop within short duration. Compared to conventional plant breeding, it takes around 3 to 7 years, but in mortar selection we have only gone for 2 to 2.5 years. Social impact. Due to salinity, majority of the farmers were not able to cultivate the crop properly, so they left the cultivation of crops because of salinity. So we, through the development of saline tolerant genotypes, the farmers can produce those crops and which can ultimately improve the farmers' livelihood. This technique is eco-friendly because we have utilized pot and trays, so we don't affect the environment. And another one is it ultimately increased the farmer's yield because around 6.8 million hectares of land were affected by salinity, which lands can be utilized if we develop the saline tolerant genotypes. So the production can be increased, rice production can be increased in future. So we can ultimately meet the demand of rice for the population. These are the reference we have utilized for this study. Thank you. EC meters, which shows presence of salinity.
Whether you measured in your field?
Yes, sir.
How much value?
In coastal area it is around 2.8 to 3, sir. Here in Faculty of Agriculture we have measured it about 2.8 with a pH of 7.9. Currently it is 2.8. In future it goes up to 4.5. Normally rice crop can't grow easy about 2, sir, which tends to go on saline condition. So the crop need, if the crop need to grow on saline condition, we need to improve the genetic nature of the crops. Then only we can meet the demand of food for the futures. Currently we are sustainable with the food production. But in future, it's not going to be like that. So.
My name is Prabhakaran.
தம்பி ஒரு நிமிஷம் இப்போ ஒரு ஃபோட்டோ எடுத்து தம்பி கீழே சார்
Good evening, everyone and all. My name is Prabhakaran, full-time research scholar from Department of Manufacturing Engineering and Technology. This is Hari Prasad, another research scholar who have worked in this project. Our project title is Indigenous Design and Development of Low-Cost Energy Efficient Additive Manufacturing Technology using Cold Metal Transfer or Welding Technology. We have done this project under the guidance of Dr. P. Sivaraj, Associate Professor, and Dr. V. Balasubramanian, Professor and Director, Center for Material Joining and Research, Department of Manufacturing Engineering. Our intention of this project is to develop a technology which can affordably and also can enhance the base material property or the product which is developed to a higher strength level and cost benefit as well. So we have chosen the material called aluminum 5 series, which have a major alloying composition of aluminum and silicon, which is widely used in automotive, aerospace and mechanical and defense technologies. This component based on aluminum is widely developed using casting process, and then if it is required, it will be gone for machining also. So during casting, the material will be melted down, and during machining, the material will be subjected to some sort of stresses during the CNC. So what happens during the heating processes, the alloy segregation will happen. When the alloy segregation is happening, the precipitates will form, which will weaken the structure. And during the machining, even if we use high cost CNC machines, what will happen is structural stiffness of the machine and dynamic accuracy and vibration will induce some stresses in the component. So once it is after come out of the machining, it will also have the residual stresses which will affect the real-time performance of the component. So we have adapted to a technology called additive manufacturing. In that particular, where additive manufacturing is used. Why additive manufacturing uses, though it is developed 30 years before, and it has gained more importance because of its advanced processing and low cost efficiency. What is the additive manufacturing is, we can deposit the metal by layer, and we can consolidate a component which is very near to the required shape. Okay, so it has better geometrical freedom, and it can shorten our production time, and we can improve the production volume, which is very important for our manufacturing sector, which also because it reduces the number of steps. We have to design the structure in a software, and we have to input that to the system, and the system will do the rest. But what we have done is, we have done the component in a semi-automatic manner. Okay, the wire will be fed, but we have to create the required geometry. So this is why we have chosen the aluminum, why we have chosen this product process. Next is aluminum have a wide variety, and different researchers around the world have developed different components. Okay, this is one second. This is a rocket shaped nose cone and with fins. This is impeller for turbine, and this is a complicated shape which we cannot easily get through by casting process. This is a cone, and this is a wing spar which is used in aeronautical applications, and this is a wide structure which is created. Okay, this is a work which has been done over this recent years in aluminum. And our origin of the problem is, though we have chosen the additive manufacturing process, there are different processes which will aid to develop the components. In that, there is something which is based on powder metallurgy and something which is based on metal additive. In powder metallurgy, the cost of the machine itself very high, but the bed size which is used to create the component is very less. Okay, so we cannot create larger components. That is the thing. And moreover, if we create a component to powder metallurgy, there will be something called surface undulations. The surface roughness will be there, so it cannot withstand higher performance environment. So we have chosen the wire arc additive manufacturing, which is a form of additive manufacturing, to enhance the strength of the component as well as the feasibility of production of that component in the cost base as well as deposition rate base. Our solution, like I said before, we have used two technologies, method of depositing the material. One is cold metal transfer, which have given the best result. We cannot restrict ourselves within the cold metal transfer method alone. So another wire arc material also we wanted to clarify whether the chosen process is having better performance or not. So we cannot say the process alone we have chosen a better performance. So we have chosen another process to validate our process selection. The objective of the work is defined here. We have used aluminum magnesium filler wire, which is a 5 series aluminum alloy. We have studied tensile hardness and impact strength, and we have identified the microstructural features through SEM and EDS. This is the chemical composition of the wire. The wire we have used is ER5356. The base metal property of the wire is given here, which has a maximum ultimate tensile strength of 250, 200 to 250. So, and the elongation around 10 to 15. And these are the process parameter we have chosen for the study. One is for GMAW process, this is the process parameter, and CMT process, this is the process parameter. The one thing that we have to look over here is the heat input, which is 0.313 for GMAW process and 0.243 for CMT process. In this, we could clearly see that the CMT process has low heat input. Like I said before, during the casting process, the aluminum has to be melted down. So during that, alloy will be segregation will happen. That is because of more heat will be generated during that process. Here, by nature, the process itself produces lesser heat. So this process, by default, we can say it can produce better results. And this is the experimental setup that we have in our center, C major, which is available in the Department of Manufacturing Engineering Technology. This is the power source, which is the entire setup is Fronius machine. This is the power control module. This is a rotating table to create the cylindrical component, and this is the virtual component that we have created during the study. This is the actual video of the component. While creating the component, the table will be rotated and the filler material will be melted down and it is deposited layer by layer, that you can see from this video. Please take a minute to look the video. You could see the circle which has formed. There is around 2 layers of deposition has been happened. We have developed a component which is around 160 mm, that is 16 centimeter. This is the actual height of the component, which is produced by two methods. One is GMAW process and another one is CMT process. And this is the diameter of the component which we have developed. This is around 90 mm internal dia and 7 mm thickness. This is the component produced after machining. The view of the component after machining for both GMAW and CMT. And this is the photograph of smooth and notch tensile specimens. And these are the tested specimens before and after the testing. These are all the notch tensile specimens and after tested specimen. These are all the impact tensile impact specimens, and these are all the image of tested samples. All the testing were carried out according to ASTM standards. In GMAW process, by visually we can see some nodules and rough surface, but in CMT process we cannot see that because the process is very smooth and clean. Next is this is the tensile property which we have evaluated. We have evaluated the component in two regions. one is at the bottom as well as at the top. Half of the component is considered from the substrate, it is to the middle, it is called bottom half, and the remaining is considered as top half. So the tensile sample have evaluated and it is recorded for the CMT component. The black and brown curve shows the CMT component, which shows higher tensile value as well as higher elongation, and in from GMW process we can see the strength of the component is slightly reduced. We will explain the reason later. And here we could see in the top region the strength of the component, the sample is coming around 258, which is higher, 8 megapascal higher than the base metal property, and the elongation which we have obtained is around 49.2%, where in the base metal it is around just 10 maximum 10% of elongation. So maximum elongation is obtained. So when comparing between the CMT and GMT, we could see the elongation is higher only for the CMT process, and we could see from the North stress ratio the ductility of the component is higher for the bottom region, and this is the hardness value that we have plotted for the CMT and GMW process component for the both top and bottom regions. For the GMW process, again the hardness value is lesser when compared to the CMT process in both of the regions. And in impact strength also, the CMT components have performed well. And this is the porosity analysis. During the production of the component, even in additive manufacturing process, what are the process? The formation of porosity is inevitable. So we cannot completely neglect the formation of porosity, but we must have a better control over it. So with this process, we have identified, we have controlled the formation of porosity. So here you could see GMW and CMT for both and top regions. This image is taken by microstructural analysis. First the sample is taken out from the produced component and it is polished and etched, and we have found out pores. Then it is given to the image analysis software. The software named as Image A, where we have pointed out the locations of the holes. Then the using some threshold, we have only have the better look of the porosity alone. Through that we have calculated the number of pores in the sample and the number of what is the total area of the pore and what is the pore diameter. In both CMT components, the top region, between the CMT component, the top region produces lesser number of pores when compared to the CMT component. When compared between the process, the CMT components produces less number of pores and pore diameter, pore area. So this is the reason for the increased strength of CMT component technically. And we have also identified the went for the SEM analysis for both the CMT and GMW process. As I said before, the magnesium will segregate and it will form precipitate. So actually magnesium is a solid solution and strengthening alloy. When it is completely dissolved in the aluminum matrix, it will provide strength. But if it takes more time to segregate, then it will form precipitate. So in both this component CMT and GMW, the CMT component produces less volume of less mass percentage of magnesium, which means more amount of magnesium has completely dissolved into the aluminum matrix. That is also the reason for the higher strength in the component. This is for the top region. In the top region also, the CMT components have less volume of magnesium alloy. And since we have produced the component, we cannot restrict ourselves that methodology that we have carried out alone have the better results. So we have went for the comparison between other researchers who have done the work in aluminum material. Here the casted alloy have 250 and other casted property of the alloy is represented by the author is 202. In our case of CMT, it is higher than all the cases, which is 254 for the bottom region and 258 for the top region, and the elongation is higher for the top region only. Okay. And when comparing between the process of additive manufacturing, okay, that is between the methodologies, and this is between the process for laser powder. These two are powder metallurgy based additive manufacturing. For laser and electron beam and CMTR, this is the comparison that we have given for the, in all the cases, say deposition rate, power efficiency and cost, the CMT component have 10 times higher rate of deposition, which means 10 kg of component can be produced within an hour, and the power efficiency, it only take lesser amount of power to produce the component when compared to the other process of additive manufacturing, and as well as the cost wise there is a 70% decrement can be achieved in the CMT process based components. Actually this is the reason for the better strength. The lesser cost deposition has been explained in the previous slide, but the reason for the low heat input, because that is the main reason for the higher strength of the component and low volume of magnesium in the matrix. So the reason is, here you could see there is a video here. You could see there is a filler wire which is depositing the material. Okay, you could see the movement of the reason for the development of lower heat input. What happens is, when the filler wire touches the base material, which means the substrate, the heat input reduces nearly to the half. So, and when it moves above from the base material, again because of the generated heat input, the filler wire will melt down, and when it is moves away from the base material, it will deposit in the layer, in the substrate material. So this process will continue. So nearly half of the current input will be reduced. Because of that, the heat input of the process will be reduced in this type of welding process. Okay, so that is the major reason for the reduced heat input in the process, which enhances the strength and other properties of the component produced with this process. So this is the thing that I have discussed here. Conclusion: the UTs of the CMT process component are 18.13% higher in the bottom region and 15.17% higher in the top region compared to the GMW process, and the elongation percentage obtained in the bottom region is 31% higher and 91% higher in the bottom and top region respectively when compared to the GMW process. And when comparing between the processes, we can conclude that the CMT process have better results than the CMT component. So that is the end of the slides. This project is funded by Department of Science and Technology, Ministry of Science and Technology, Government of India, New Delhi for the financial support rendered through fellowship under first phase 2 scheme. The author wishes to record sincere thanks to Miss Fronius India Private Limited, Chennai for technical support. Thank you. No, the current is not constant, sir. Initially, the current will be higher. That is, this has two spaces, arcing phase and short circuiting phase. During the short circuiting phase, the filler wire will come in contact with the substrate, and there will be short circuiting there. During the short circuiting, the current curve, you can see, it will be reduced to half of its value. So because of that, there will be heat generation, but it will not be more than the GMW.
How you measure this?
So this is a graph which is provided by the Fronius. Okay, because they developed this technology. This machine is we have purchased from Fronius. So this is the
manually you take this one.
Yes, sir.
Manual of the machine.
Yes, sir, manual. So when it is moved away from the base substrate material. Because of the generated heat, the filler material will melt down and it will start deposit. So that's what actually happens in the additive process.
ಇದು UPS ಇಲ್ಲ ಫ್ಲಾಟ್ ಮಾಡಿದ್ರೆ ನಾನು ಓಡ್ ಮಾಡ್ತೀನಿ ಏನಪ್ಪ ಇನ್ನಿಂದ ಬೇಗ ನಮ್ಮ ಇರುತ್ತೆ
So I'm Shubham Singh and this is my teammate Deepa Swati Lakra. Our project was detection and drug profile analysis of circulating tumor cells in multi-drug resistance cancer model. So basically this whole concept is based on the existence of circulating tumor cells, which are cells which get detached from the primary tumor cell mass and flow through the body. Throughout the different stages of cancer, these cells keep getting separated, and as the tumor progression takes place, the higher the stage, the more number of cells would be present in the blood. Also, these cells are the prime reasons for metastasis through which secondary tumors are formed. So these are a very important cell, and research on them is very important for, we believe, for any further development. So our main objective included these three aims, like screening of blood sample for detecting and isolating CTCs. We aim to use these two techniques, collagen adhesion matrix and immunofluorescence techniques. In vitro drug profile analysis based on patients' drug profiling, which drugs they are to use. We would also treat the cells with the same drug combinations and histopathological analysis on CTC-based animal models. So this is the primary flow chart we prepared. So after categorizing patients in different groups through sex, age, type of cancer they have, we wanted to collect blood at different time intervals, and at all those intervals we will detect what is the general CTC count in the blood, and depending on whether the treatment, because we will do the follow-up checkup also, depending on during the follow-up whether the count increased or decreased, we could predict whether the therapy was going in a proper direction or not. If the count increased, that means despite the drug treatments, the drug's efficacy was not enough to suppress the cancer, which means we could suggest the doctors that okay, this is the current status, and even after, let's say, 3 or 6 months, the CTC's count is not increasing. So we may pursue giving different type of drug combination treatments. This is again a simple flow chart. So we'll take from population, we'll take the blood samples through conventional immunofluorescence and CAM assay. We will detect the CTC, and through CAM assay we can select the viable circulating tumor cells which we can culture for our in vitro studies. Now this is for the animal model. So here what we take, we will get a new mice, and through the CTC we culture, we will incorporate our cancer in tumor generation in them, and we basically divide our mice in 5 groups: negative control, positive control, and the other 3 groups would be based on the drug combination. 2 would be single drugs, and 1 with the combination of drug. And based on that, as you can see on the screen, at different days, in a very programmed way, where on the 6th day tumor would be induced, at the 20th day treatment will start, and then at the 30th day histopathological analysis. For histopathological analysis, the rats would be killed and the samples would be taken. We programmed complete this flowchart. These are the expected outcomes of our studies. So first is the therapy response based on CTC count, whether, as said before, based on whether CTC count is decreasing or not, in the follow-up treatments, we can detect, predict whether the therapy is going in a right direction or not. Second is personalized treatment plan. So whether based on the therapy response, whether we need any specific drug combination which might work in the said patient, we can suggest our recommendation to doctors like, okay, this drug is not working, this drug was working in other samples, we can use this drug. That's the personalized treatment plan. And third is the insights regarding extracellular matrix, extracellular microenvironment in secondary tumor establishment. When these circulating tumor cells go around the body, what factors influence them for development of that secondary tumor on that extracellular matrix level? We wanted to detect these. These are a few references for our studies. And thank you.
To be given for this one?
Sir, here we have taken like
The drug doses. This is same for all the cases or it may differ?
This we take a model drugs like paclitaxel and cisplatin, and the drug doses which we are using different scientific studies which have already been
It depends upon the patient
it
severity
also
of
depends
the
on-
tumors.
yes. Like we have taken this as an example. Let's say patient is being treated with paclitaxel and cisplatin, and based on our academic study, we have taken the range of dosage drug which can be used. Now let's say if any other patient is given doxorubicin or any other drug, so based on that we will change it. But this is like the prime. Our ethical permission is pending right now.
Oh,
So as soon as we get that, we will.
you can start.
Yes.
Okay. Next. Start.
So good noon to one and all gathered here. Sorry for the inconvenience me. And I'm Alamelu Mangamuttaya and this is VK Pavitra here to present on the title Writing Therapy in Ameliorating Mood. We are from the Social Science cadre. And the background of the study goes like both handwriting and mood cannot be remain stable for a period of longer period of time. The best example can be quoted is our answer script in which the first page and the last page won't be remaining the same. And similarly, the one more example I have given is the letter. The letter which we used to write in the olden days to convey our messages. It's one form of the expressive writing which we put all of our emotions into that letter and which will not be affected into our heart for a deeper period of time. So it will be expressed in simple. The emotions will be expressed out in an easy way. And the introduction goes like expressive therapy. Actually, the writing therapy is from the branch of expressive therapy, and it's also can be expressed through art, writing, dance, music, and drawing. We have chosen writing therapy. It is like the process of writing or processing the written information. That is what we call to be the writing therapy. The third one is the mood, which is the affective component. In psychology, we call it to be the ABC aspect, in which A is the affective, B is the behavioral, and C is the cognitive aspect. And here we chosen the affective component, which reflects the emotional state of an individual. And the scope of this study is like the first pictogram will represent the repressed emotions of individual who is having the mental health issues. Similarly, the second pictogram will represent how much the people have been using the writing therapy and they have overcome their mental health. Subjective mental health is being improved. That is the second pictogram representation. And the third, next we get into the hypothesis wherein the writing therapy has a significant impact on improving individual's mood, and there is a significant difference between the pre-test as well as the post-test in the terms of self-rated changes. Self-rated changes is nothing but how we feel about ourselves before and after the writing therapy session. The third point is that the significant difference between the individual handwriting in relation to demographic variables, as in the further slides we will be getting into knowing the demographic variables. Next slide is about the objectives, which is similar to that of hypothesis. like improving the mood through writing therapy, examining the self-rated changes of pre-test and post-test, and analyzing the apparent differences between the handwriting in relation to the or in the terms of demographic variables. This is the research methodology wherein the research design is a quasi-experimental and one group pre-test post-test method. Why we chose quasi-experimental is that we will be focusing only on a particular one group alone. We won't be having an experimental as well as a control group. We will be only focusing on one group, that is what one group pre-test post-test method. And the sampling technique is purposive sampling, as we purposively will be focusing on the mood disorders. And the sample size is 50 and the sample group is people with a mild neurosis. Neurosis is a condition where the individual will be having an insight of what they are being suffering from. Say for example, in a medical condition, if I say I am having a fever, I will be knowing that I am having a fever. Excuse me. And similarly, in a psychological state, if you are having a neurosis condition, we will be knowing that yes, I have been affected from stress, I have been affected from depression, so and so, like that we call it to be the neurotic state. And the intervention goes like writing therapy. And these are the variables of the study. Independent variable is the writing therapy. Dependent variable is the mood. Demographic variables are age, subjective emotional state. Subjective emotional state will represent what sort of emotion that an individual is having before going into the intervention strategy. And the duration of the repressed emotion is that how long the emotion is being suppressed inside the individual, and the number of attempts to overcome, like how much time they have get into the medications, all such things will be demonstrated. This is the sampling framework wherein the screen sample will be 50 and they will let into the explanations of procedure of recalling the repressed emotions and obtaining an informed consent. In psychological or the social sciences, it is a it's mandated to get informed consent from the individuals as we are getting their writing materials. And the third step will go like pre-test of the subjective self-rating, intervention of writing therapy, post-test subjective self-rating 1 to 10, and analysis of mood and handwriting. The analysis part is divided into two types, where is handwriting analysis and statistical analysis. Handwriting analysis is the evaluation of the writing samples done by the graphologist with the help of proceeding with the clients, and statistical analysis focusing on the paired sample t-test. This is the writing sample in which we when we compare the first and the last few lines. The first line will be depicting a clear handwriting, whereas the last will be a congested or illegible ones. This will denote the disrupted flow of emotions that an individual is having. And the ink blots at the specific point of efforts and without will denote that they have been struck up when they are being writing things. So this is the graphical representation, the before and after the writing therapy in the case of anxiety, stress, depression, guilt and grief. The expected outcomes are like to ameliorate the writing therapy in ameliorating individual's mood, subjective symptom relief, and creating awareness on the mental health professionals, especially on the expressive writing therapy. The limitations are like the writer may experience the emotional outbursts on bringing back the suppressed emotion, and the study cannot be implemented to the neurodevelopmental disorder cadre as it is they cannot be connecting themselves. The scope of commercialization is that this is similar to that of cognitive behavioral therapy, and it is one of the cost and time efficient strategies to deal with mild neurotic cases. Thank you.
What is we using in the last?
We can use it in the hospital setups.
Hospital setups.
Yes, sir.
You are concentrating on the strokes of the right, or what is the thing? How you are going to verify the mood? For example, you told that in the beginning and the end, you are told that there is some change in the
Yes.
writing. For example, instead of manual visualization, I have to maybe computerize this.
Okay.
Means what are all the parameters you are going to consider it for checking the mood?
So when in case of graphology, I am a graphologist, I can assess the person with the handwriting. So we
Okay.
can't predict, like we can't manipulate, we can't make it a machine thing, because each individual will have a different strokes and
okay.
different things.
Ah.
So we can't computerized things. Like if I say A
Is it
plus B,
possible to incorporate the artificial intelligence for these things?
we can, but the studies are still in process
Going
on.
on?
Yes, sir.
Because
It's
by
not
using
evident.
the AI, you may incorporate this means it's very cost effective and anybody can use it.
Yes.
Not by using the person.
Yes.
Anybody can just, I'm scanning the letter of the skin, even in school students or anybody else, for
But the
example.
point is that if a normal individual, when they are given to write things on their own without the help of a psychologist or a mental health professional,
Okay.
it can trigger out the negative emotions more than the positive
Yes.
emotions. So it is mandatory that you need a psychologist or a mental health professional.
That is additional support means you can use that
Yes.
also.
Mm.
Yeah, very nice idea.